Hello, welcome to my channel Correct Biology. My name is Dr. Vikash Bhardwaj, and in this video we are going to discuss what is the difference between DNA and RNA. So here I am going to teach you the basic difference between DNA and RNA. Be with me and learn the difference between DNA and RNA. Now here, before starting the video, I want to ask you one simple question. Very simple. Try to see here. Let us suppose you are going to have single-stranded DNA. This is single-stranded DNA sequence 5' AT GC TA 3'. You know very well, A here stands for adenine, T for thymine, G for guanine, C for cytosine. So these are the base which are present within the DNA, and this is single-stranded DNA. Now let us suppose you take out all the thymine and you replace thymine with uracil. So this is single-stranded DNA. From this single-stranded DNA, if you take out the base thymine, and here if you add in place of thymine uracil, so here this will be the sequence 5' AU GC UA 3'. Now here I just want to ask you, this sequence, is it DNA or RNA? What is? Is it DNA or RNA? Confused? Okay? Yeah, I know you must be knowing the right answer. Anyhow, I will tell you what is the right answer. Just learn the basic point, the difference between DNA and RNA. Now here I just want to tell you, you must be knowing nucleic acid, it can be of two types. One is DNA, and here you must be knowing DNA, it stands for deoxyribose nucleic acid. In case of DNA, the sugar, here you see deoxyribose, here the sugar is going to have at the 2' position hydrogen atom. Keep in mind that in case of DNA, the sugar, which is present within the nucleotide, it is going to have at the 2' position hydrogen atom. While in the case of RNA, what you are going to see, if you expand the RNA, it is ribose nucleic acid. In case of RNA, the sugar which is present, it is actually the ribose, and here what you are going to see at the 2' position hydroxylic group. So this is the basic difference between DNA and RNA. Now here I just want to tell you, if you see DNA, it is actually the polymer of nucleotide. Hopefully you must be knowing what is nucleotide. Base plus sugar plus phosphate. I have told you, in the case of DNA, the sugar is deoxyribose. Now DNA is a polymer of nucleotide. We have discussed the structure of DNA in great detail. So here you can see that DNA, it is polymer of nucleotide. You can say it is a polymer of DNTPs, deoxynucleotide phosphate. Adenine, thymine, guanine, cytosine, they have been linked together. Now here in the case of DNA, I have told you at the 2' position of the sugar, you are going to have hydrogen atom. While if you see in the case of RNA, RNA is polymer of ribonucleotides. You can say it is a polymer of RNTPs. Now in the case of ribonucleotides phosphates, what you are going to see at the 2' position of the sugar, you are going to have hydroxylic group. So this is the basic difference between DNA and RNA. Now in the case of DNA, you must be knowing DNA is the genetic material in most of the organism. DNA is going to store genetic information, and DNA acts as genetic material in most of the organism. While if you see RNA, generally it do not act like genetic material, but in certain cases, like in some RNA virus, what you are going to see RNA as the genetic material. most of the time DNA, it is going to act like genetic material, but in certain cases RNA can also act like genetic material. Now here, if you see the base composition within DNA, hopefully you remember the structure of DNA. A is going to pair with T, adenine is going to pair with thymine, guanine is going to pair with cytosine via hydrogen bonds. Between adenine and thymine, you are going to have 2 hydrogen bonds. Between guanine and cytosine, you are going to have 3 hydrogen bonds. So in DNA, generally what bases you are going to see? Adenine, thymine, guanine, cytosine. Yes, here hopefully you remember adenine and guanine, they are purine. Thymine and cytosine, they are pyrimidine. Now here, if you see in the case of RNA, generally what you are going to see, in place of thymine, you are going to see uracil. In place of thymine, you are going to see uracil. So in case of RNA, generally RNA is single stranded, and what you are going to see, RNA, it can fold on itself. In that way, adenine is going to pair with uracil. Guanine is going to pair with cytosine. In case of RNA also, if it is going to fold on itself or it is double-stranded RNA. Okay, generally RNA is single-stranded, and generally what you are going to see, DNA is double-stranded. But you can see also single-stranded DNA also. Now here I just want to tell you, DNA generally it is double-stranded, RNA it is single-stranded. Now here let us come to our question. Let's suppose you are going to have this single-stranded DNA. The sequence is 5' ATGC TA 3', and let us suppose you replace all the thymine with uracil. Okay, now will it become RNA or it will remain DNA? Here I just want to tell you the right answer is it will remain DNA. Why? Still here in this sequence the sugar is deoxyribose. Keep in mind that in case of DNA, sugar is deoxyribose. Even you replace thymine with uracil, okay, still this is going to remain DNA. Why? Because here the sugar is deoxyribose. So here the sugar is going to decide. If sugar is deoxyribose, it is DNA. If sugar is ribose, then it is RNA. Hopefully this is clear to you. So this is going to remain DNA, deoxyribose, because here the sugar is deoxyribose. Now here I just want to tell you, yes, here we see another important thing about DNA, that is DNA is stable under alkali hydrolysis. Means if you are going to have DNA and if you are going to heat the DNA in the presence of alkali, in the presence of NOH, if you heat the DNA, it is going to remain stable. Yes, if it is double-stranded DNA, it will get denatured, the both strands they will separate, but upon cooling it will again renature. Nothing is going to happen to the DNA. DNA is stable. So keep in mind, DNA is stable under alkali hydrolysis. Why? The answer is simple. In case of DNA, the sugar is deoxyribose. At the 2' position, you are going to have hydrogen atom, and that hydrogen atom will not attack on the phosphodiester linkage. Okay, so in case of DNA, at the 2' position, what we are going to see, hydrogen atom, and this hydrogen atom will not attack on the phosphodiester linkage. So DNA, it will remain stable when you are going to heat the DNA in the presence of alkali. But here if you see the situation, RNA, if you are going to heat RNA in the presence of alkali, what will happen? RNA will be degraded. So if you are going to heat RNA in the presence of NOH, RNA will be degraded. Why? The answer is simple. In case of RNA, so here I have shown you that in case of RNA you are going to have base, then sugar, then it is attached to the phosphate group. You are going to have this phosphodiester linkage. Now here, in case of RNA, at the 2' position of the sugar, you are going to have hydroxyl group. So this hydroxyl group, which is present at the 2' position, it will attack on the phosphodiester linkage. What will the result? The phosphodiester linkage will be broken. So RNA will be degraded in the presence of alkali, but nothing is going to happen to the DNA. So keep in mind, RNA, it will be degraded under alkaline condition. When you are going to heat RNA in the presence of alkali, it will be degraded, while nothing is going to happen to the DNA. DNA will remain stable. Answer is simple, because in the case of RNA at the 2' position, you are going to have hydroxyl group, and this will break the phosphodiester linkage between the ribonucleotides. So hopefully this is clear to you. Now another important point here, I just want to tell you, in case of DNA, you are not going to see any enzymatic activity. In DNA, till date, no enzymatic activity have been reported. Okay, so DNA is not going to have any catalytic activity. Here you are not going to see any enzymatic activity. But in certain RNA, not all, some of the RNA, they can have catalytic activity, they can have enzymatic activity. And hopefully you know, the RNA which are going to act like enzymes, they are known as ribozyme. They are known as ribozyme. Yes, we will have discussion related to ribozyme in another video, but keep in mind, RNA may have enzymatic activity, but DNA, it is not going to have enzymatic activity. So these are the some basic difference we have discussed related to the difference between DNA and RNA. Hopefully you have learned the difference. Please do like, share the video, and subscribe to my channel, Correct Biology. Correct Biology.